I think this.
Okay. Kia ora everyone, and thanks for coming along to our digital event today. I'm just going to kick off this event with some housekeeping slides for those who haven't joined us before for an online event. We like to start off our events with whakatauki, and this one in particular sits above the desk of our head of researchers in our head of researchers office, and it goes ehara, takutoa, ite toa, takitahi, engare, he toa, takitini. And that translates to my strength is not as an individual, but as a collective, which is something we find really resonates with the research community and at the foundation. As you can see, your camera and microphone have been automatically turned off, and you won't be able to turn these back on for the duration of the event. That means that we can't see or hear you. But if you would like to communicate with us during the session, you can use the chat and Q&A functions. You'll find them at the bottom of your screen. They're labeled chat and Q&A, and if you just click on those icons, it will open up a small box and you can type your message in there, and you'll see that it is set to send these to myself and the panelists. So you can use the chat box to ask us technical support questions, and we'll do our best to help you. At the end of Dr. Anchan's event, presentation today, we'll be opening up for a Q&A. So you can use the Q&A function to send through any questions that you might have at that time. It's also down the bottom of your screen, so you just click on it, open up another chat box, and you can type your question in there. All the questions will be visible by all the attendees. However, you can tick to send your question anonymously if you want to. Once it is submitted, we will either opt to answer the question live, or we might type a response to you. Please keep in mind that we have a limited time frame, so we might not be able to answer all of the questions. Please keep all of the questions to the topic presented, and our speaker won't be able to answer any personal medical related questions that you might have. At the conclusion of this session, the Zoom will end for everyone. It can be a little bit abrupt, so my apologies for that, but please do keep an eye out for the recording, which will be available towards the end of this week or early next week. Okay, so now I'd just like to take a moment to introduce Dr. Akshata Anchan, and that is our speaker today. Dr. Anchan has a PhD from the University of Auckland. She is a postdoctoral researcher and is funded by a Neurological Foundation First Fellowship. And Dr. Anchan specialises in how invasive cancer cells cross the blood-brain barrier. So Dr. Anchan is joining us today from the Centre for Brain Research at Auckland University. So thank you very much, Akshata, for coming along and presenting today. And now I'll hand over to you.
Sure, thanks. I'll just share my screen quickly. Cool. So I hope you guys can see that now. And so hi, thanks for coming for our talk today. And thanks also to the Neurological Foundation for organizing this event. So I'm Akshata, and I've just completed my PhD 2 years ago, and I'm now working at the Centre for Brain Research, looking at melanoma, which is a skin cancer, but particularly focusing on how it gets inside the brain by interacting with what we call the blood-brain barrier. So before we get into the actual talk, I thought I'd spend some time just sharing my background and letting you all know how I got here. So I was born in India, which is a totally different world from New Zealand. But when I was about 10 years old in 2005, I came to New Zealand. And since then, which this means that I've done all of my primary and secondary school learning in New Zealand, and I graduated from high school out west in Avondale College in Auckland in 2012. And I just want to note that even at this time, all of the subjects that I really, really enjoyed were always around science. And because of that, I decided to pursue my education at the University of Auckland, in biomedical science. And biomedical science is sort of like a research-based degree because you essentially get introduced to a whole range of topics in medical and biological sciences. So I was very interested in that, and I graduated in 2015 from UOA with my undergrad degree, but decided to continue to study because I really enjoyed the papers that I'd done in undergrad. And what I'd found was that I was really, really attracted to the topic of neuroscience, but also of cancer biology. And I sort of couldn't pick which one I wanted to work with at this stage. And that's when I was lucky enough to found a research project that perfectly fit both of those interests. So I went to a networking event where I found my supervisor, who was essentially trying to make a project looking at cancers that specifically migrate inside the brain and then become brain cancers. And I thought that sounded extremely interesting and also an extremely important topic to research. And that's how I built my project back in 2016. And so my research project is essentially looking at invasion into the brain by melanoma, which is an aggressive skin cancer, as a lot of us probably already know. And I'm specifically looking at how this cancer that originates in the skin gets inside the brain from the skin. And most of this project was done predominantly at the Center for Brain Research at the University of Auckland. And just a little plug here where, you know, like other PhD students and researchers, while we do a lot of work on the research project itself, we actually also spend a lot of time volunteering and doing a lot of other events where we discuss the kind of work that we're doing to the general public and also to academia. So because of that, I was quite interested in the research that I was doing, and I completed my PhD in 2021, which means I'd spent quite a few years on this specific topic. And by this stage, I was pretty certain that this is the research field that I want to build my career into. And because of that, after I graduated with the doctorate, I decided to start my postdoctoral research, which was granted and funded to me by the Neurological Foundation First Fellowship. So now we can get to the actual talk. So my talk is looking at migration of brain metastatic cancer cells across the blood-brain barrier. Now this sounds quite long and complicated, but I can sort of try help break it down. So a metastatic cancer is essentially a cancer that can leave the site where it originated. And just give me a second, I'll try to get a laser pointer up. Here we go, yeah. So metastatic cancer is essentially a cancer that can leave the site where it originated. And then for melanoma, that would be at the level of the skin, and then travel to any other part of the body. So when we say brain metastatic cancer, a brain metastatic cancer is one that specifically metastasizes to the brain. And the aim of my project is to identify any kind of entity or proteins that are expressed by brain metastatic cancers that help their migration into the brain. So there are actually several different types of cancers that can metastasize to the brain. And of these, the three most likely cancers are lung cancer, breast cancer, and melanoma, which is skin cancer. However, what's interesting is that of these three, it's actually melanoma that has the highest propensity to sort of specifically and particularly migrate to the brain. And that's what a lot of my PhD was actually based on. And hence, a lot of my previous studies were also conducted primarily with melanoma cells as opposed to lung or breast cancer. So here's a little bit of background on melanoma and its migration into the brain. So as we know, if we spend a lot of time out in the sun, we're exposed to what we call UV radiation, which comes from the sun. Now, if we spend a little too much time in the sun and we didn't put a nice coating of sunscreen on, then we could develop some moles on our skin. Now, usually these moles are benign and okay and not problematic. However, at some point, one of these moles might start proliferating or dividing, and it might form something like a tumor at the level of the skin. Now, if this tumor at the level of the skin starts growing and starts producing these projections, these little fingers that come out, what that does is it can actually find a blood vessel at the level of the skin. Now, if the cell was to try and invade and intercalate into the blood vessel, what that means now is that suddenly it's got access to go to anywhere else in the body, because your blood vessels in your circulation goes to anywhere in the body. So the cells can now go from the skin to anywhere else. And what we find with melanoma, as I had said earlier, is that melanoma seems to preferentially go from the skin into the brain. However, to do this, the melanoma cells in the blood would have to actually squeeze back out of the blood vessel at the level of the brain and then form a secondary tumor there. And so what we find is for brain metastasis to occur, for a cancer to actually get inside the brain, there are two main parts that we need to consider. The first one is for the cancer cell to get out of the blood vessel at the level of the brain, which is part one. And the second one is for it to actually survive in the brain once it's gotten through. And that kind of makes sense because the brain is a totally different environment from the skin. So we need to consider how the cancer cells actually survive here in this new environment. So a lot of my PhD was actually conducted primarily on part one. And so most of my talk today is going to be for this part. So as we said, melanomas from the skin typically try to travel to the brain. And it's a little bit interesting because the way that this happens is through the circulation. Now, as we know, the brain is an extremely energy demanding organ. It needs a lot of oxygen, which means that it has a lot of blood vessels surrounding, as you can see over here, to provide it with that blood flow and then that oxygen. So if we zoom into the brain over here, this is kind of showing just how many blood vessels are circulating, are enclosing the brain. And if we were to take a knife and just cut a cross section through this blood vessel right here, and look at it sort of front on, we would see something like this as a diagrammatic representation, where we've got the blood vessel over here, and the blood flow would be in this atrium just here. And at the level of the brain, we would have other cell types that are here to protect and support the blood vessel. But you can imagine that if we've got a cancer cell or a melanoma cell in the blood, this vessel wall lining is going to be the first physical structure that that cancer cell comes into contact with. And what's interesting is that this physical structure right here, which we're just going to call the blood vessel cell wall, is very tightly adhered at the borders by extremely tight junctions. So if we zoom into this area over here, what we've essentially got is one side of the blood vessel wall here and one side of the blood vessel wall here. The blood flows in this side, and obviously there's the brain over here. So you can imagine that that blood vessel wall essentially acts as a barrier to anything that's in the blood from getting into the brain. And what we find is that at the walls of these blood vessels, we've got a lot of these proteins interacting with each other really, really tightly to hold that wall extremely tightly together and essentially stop anything that's in the blood from getting inside the brain. So we can imagine that if there was a melanoma cell in the blood, it would then have to find a way to disrupt these proteins and then get inside the brain by disrupting this barrier formed by the blood vessel. And so what I'm sort of interested in is what is expressed by these melanoma cells, maybe they're proteins or maybe they're secreted items that allow them to interact with and then break through this extremely important blood-brain barrier. And I found during my PhD that firstly, that melanoma cells, so skin cancer cells, negatively affect those cells that make up the brain's blood vessel. And I found this by when I take human melanoma cells derived from patients who've had melanoma and add them to those blood vessel cells, they decrease that barrier integrity of the blood vessel cells. where say if I've got a blood vessel cell here, here, forming a nice tight wall through those junctions and the proteins in the junctions very tightly adhered cell to cell. If I add melanoma cells to it, you essentially get all these holes occurring in that barrier so that the melanoma cell can flow through, which is really not good. And this is the kind of thing that we want to try and avoid. How did I discover this? It's a little bit complicated, but I'm going to spend some time explaining how it is because it's actually quite neat and it's a neat piece of equipment that we've got. This is what it looks like. It's a computer attached to an even smarter computer in a box. And to this, we add a little plate. And the system overall is called electric cell substrate impedance sensing, which is a mouthful, but we just call it ECIS. And it can do a lot of things. So as you can see over here, essentially what I've got is this plastic plate, this rectangular plate that looks like this, and we attach this to the computer. And in the plate, we've got a bunch of these wells, and if we sort of zoom into this area over here, we've got these little compartments and wells, and we can actually put live cells inside them. And you can see that each of these wells are lined with gold electrodes, which we can put the live cells on top of. And because we've got electrodes here, what this means is that we can use electricity and current to look at cell behavior. And we can actually analyze changes in the cell by interpreting any change in the current. And what happens is that the current in the well, it actually changes according to what is in the well. For example, if the well is completely empty, then the current stays the same. It doesn't get altered at all in any way because it's an empty well. Now, if you take a cell that lines the blood vessel at the level of the brain and you add it to the cell, what happens is that those blood vessel cells form their typical barrier, their strong barrier by attaching to each other. And what this does is it impedes the flow of current. Current can't physically go through those cells. So in the system, the current drops and we can record this in the computer. And because the current drops, alternatively, what happens is the resistance to that current flow increases. Now, if we take a melanoma cell and add it to the same well, what happens is that the melanoma cells pokes holes all over the blood vessels, and then the current can easily flow through, which means the current increases, and alternatively, and similarly, the resistance therefore decreases. So this is just to summarize this is how we sort of look at it. So if the well is empty, the resistance to current flow in that well is unchanged. If the well has blood vessels forming inside it, then the resistance to current flow increases because we get a nice strong barrier. And then when we add melanoma cells, that resistance to current flow is entirely diminished by the melanoma, and so the resistance decreases. And this is what we interpret as a disruption of that strong barrier integrity. And just to note that this is actually a really neat system, because we can use current, and current is fast and extremely sensitive. So that means two things. One, we can look at things happening in under a second. So we get information almost as soon as it happens, which is great. And furthermore, we can actually detect really, really small changes because of how sensitive current is. And that's really important because we need to remember that cells are very, very small, which means we need to be able to detect minute changes. And so this is the kind of graph that we get out by using this computer, where we essentially measure that resistance in the well. And when we have, so the resistance sort of increases as the blood vessel forms. And then when you add just the blood vessel cells, the resistance stays strong and high at around 900 ohms. And then when you add the melanoma cells, you can see this fast decrease in that resistance, showing that we've completely disrupted that barrier formed by these lovely brain blood vessel cells. Which is all great, but can we actually see it? Graphs are all informative, but can we actually visually see what these cells are doing? And the answer is we can. So over here in green, I've got those blood vessel cells. I've just added a color in there so we can see it visually as green. And you can see that that's one cell there, that's one cell there, that's one cell there. They're all attached to each other very, very tightly across the whole plate. And they stay like that, and they stay happy like that, forming that big, strong layer as a barrier, essentially, looking down. And then when you add the melanoma cells over time, you can see that the melanoma cells start attaching to those cells and then spreading out and sort of giving out these big, long fibers and these long sort of fingers and projections that really start pushing and affecting those cells. We can actually see it a little bit more clearly in this diagram here, where on the top, I've got the blood vessel cells stained in green color without melanoma addition. And on the bottom, I've got the same with melanoma addition added. And if you remove all of the phase images of the cells and just look at the fluorescence alone, you can see that without the melanoma cells, you've got this nice, strong barrier where each cell is sort of attached to each other really tightly. You can see it even closer over here where all the cells are right next to each other. But when you look at the image with melanoma cells added, you can see these obvious big glaring holes in that barrier, which suggests that the melanoma cell has come inside and really pushed those cells away to get through into the brain. And we don't want this to happen. I was able to find this out and state that melanoma cells intercalate within the brain blood vessel cells. And I was lucky enough to be actually able to publish this, particularly even as this diagram, which shows the melanoma cell sitting on the brain blood vessel cells, attaching to them, and then adding these projections that really disrupt that strong barrier at the brain. And so the next question was, well, what about the other brain metastatic cancers? Can they do the same thing? We've seen this with melanoma, but what about breast cancer and lung cancer? We know that these two also metastasize to the brain, and patients always come into clinic and find that they've got brain cancers developed through these. So can they do the same? Do they have the same effect on the brain blood vessel cells like the melanoma cells do? And what I found is I was able to, in my postdoc, work also with a variety of breast cancer cells on top of the melanoma cells. And I've got some images here of the cells growing in the lab. And you can see that the melanoma cells kind of look like this. They've got these long projections coming out, which kind of look like what we'd seen earlier with the green images. However, the breast cancers are a little bit more epithelial in nature. They've got they look very different to the melanoma cells. But what's interesting is that the effect on that barrier, on the blood vessel barrier, is the same. The breast cancer cells also decrease the resistance, meaning that they're also disrupting the barrier. What's important to note here is that the melanoma cells seem to be a lot worse, where that dip is much, much worse than there is for the breast cancer. But what we find is that both cancer types can disrupt the blood vessels at the level of the brain, which makes sense because clinically we do see both these cancers in the brain. So the main question then is, how are they doing this, and can we stop it from happening to produce drugs against it? And during my research, I've tried to look at exactly that. I've looked at a range of things that could help melanoma cells really insert themselves between those blood vessel cells. And these things have included primarily proteins on the surface of the melanoma cell and proteins that are released by the melanoma cell. And this is a complicated table, but the only thing I want to sort of get across with this is that this is just a limited range of a variety of things that I've assessed, every single one of these, to see if these are the proteins that cause the invasion caused by melanoma cells. And as you can kind of see, all the no's there essentially state that none of these entirely explain how a melanoma cell is disrupting that blood vessel, which is quite surprising. What I have seen is that melanoma cells secrete some proteins. I found about 2 that show some potential in disrupting those brain blood vessel cells. However, even those are not, this is published, by the way, in the papers over here, looking at secreted proteins and secreted enzymes. However, what we found was that these proteins and enzymes, again, did not sufficiently disrupt that barrier on their own, which means that melanoma cells are either using these proteins in combination with something else or using a completely different mechanism altogether. So then what else is it? This is sort of the main question moving forward, is what is it that melanoma cells are actually using? What do they need to cause that disruption? How do we find it? And can we stop it from happening? And we've considered maybe could they be carrying any kind of cargo vessels called vesicles and releasing them that allow this. So if you've got a melanoma cell, maybe they've got particles or vessels that they package with proteins that we can't see or detect, that then go in first, disrupt this barrier so that the melanoma cell can follow later. And we think we've potentially got some evidence to prove this now. Where over here, we've got some very, very, very detailed imaging showing a melanoma cell, and you can see that here in the red with these long projections like we'd seen before. These are the fingers of the melanoma cell coming out. It kind of looks like an alien, actually, sort of really intercalating and trying to squeeze in to this the brain blood vessel cells that are surrounding. And these images on the side and on the bottom here kind of show this image, but in 3D, looking down this plane, and then in 3D looking down this plane. And you can see over here that this melanoma cell has its projections going under and it's really trying to squeeze into and past the blood brain vessel cells over here. So this is what it looks like, what the melanoma cell itself looks like. But what we've noticed while we've done these experiments is that the melanoma cells also seem to secrete or release these large bulby particles that kind of look like cells, but they're not as big as cells themselves. And this is a picture sort of trying to define that. So again, over here, the really big orange cell is the brain blood vessel, and the reds are the melanoma. And you can see that these balls actually look a lot smaller than the melanoma cell itself, but it's released by this melanoma cell. And what we find is that while in the case of the melanoma cell, you can see that the cell itself goes between the two neighboring brain blood vessel cells. These small particles seem to actually go right inside the brain blood vessel cell. It's almost as if the brain cells have engulfed these particles within them, and you can sort of see it in here where it looks like it's almost being eaten by the brain cell. And what we're thinking is that potentially these melanoma cells have these cargo vessels that they secrete or release that is then taken up by the brain and the brain blood vessel cell, which then causes some kind of signaling down the line that disrupts the brain barrier and then lets the melanoma cell proper to come through. So the project, my project from here on, is actually looking at these entities, these vessels in melanoma and also in the other brain metastatic cancers like breast cancer and lung cancer, and assessing what they are, what kind of cargo they're holding, how they get inside the brain, and is it truly them that are allowing the melanoma cells proper to get inside. And then just to finish up, what I wanted to point out is, why is this important? Why am I trying to look at all these different avenues of how cancers affect the brain's barrier and how different cancers, different types of cancers affect it? And that's kind of important because different people have different cancers. Even two people with melanoma will have slightly different variations of the same cancer because it's so personalized and so individualistic. And those different cancers will affect and have different impacts on people, which we need to be able to predict, or at least try our best to. And that's why in my career, I actually want to identify if those different cancers are a possibility in different people and what that might actually mean for therapy. And we sort of need to realize that people are different and they're affected differently by cancers. And because of that, treatment against that cancer will probably work in a similar manner. So we kind of need to understand what is there before we try to get rid of it, especially because people don't like therapy, and therapy can be quite harsh on the body. And that's why it's better to sort of try and analyze all these different avenues so we know what's most likely to work in the safest way, before actually using it for therapy. And that means that we can't really do this. I can't do this all on my own. We actually need help from a variety of different angles, so we can try and cover all the possibilities of brain cancer to the best of our ability. So in our lab group, we actually have a really big brain cancer facility where, while I work on melanoma and brain metastases, we've got other researchers also working on this. And these are just some examples: Laverne, Hyunju, and Jane, who all work on glioblastoma, which is a primary brain tumor. It develops in the brain. So they're working on that and trying to find therapies against GBM. And I've had a lot of past students myself looking at my project and looking at melanoma migration into the brain. We've had Sam, who's tried to develop a 3D model of that brain's blood vessel to study invasion. Yota has also been Yota and Libby both actually have been looking at a variety of different proteins and doing functional assays to see if they facilitate melanoma invasion. And more recently, we've had Dana and Arisha. Dana specifically has helped me look at those vessels, those entities, and produce some of the really pretty pictures that I showed today. And Will, as a recent student who we've got doing a master's, who's looking at how those melanoma cells survive once they're inside the brain, which is looking at that part 2 that I talked about earlier in the talk. So as you can see, we're really trying to expand our research and try to cover as much ground as we possibly can in this field, which is necessary. But that's sort of just the lab collaborations. On top of that, we've actually got collaborations with a variety of people to try and facilitate our research. The first one who I started off with actually was Dr. Graham Finley at the Auckland Cancer Society Research Center. And this collaboration was really important because my background was at that stage with my supervisor was primarily neuroscience. So we needed someone from the Cancer Society to help us out with the melanoma lines. And of course, also through Sir Richard Fall and other members of the Center for Brain Research and the Neurological Foundation Human Brain Bank, all of which has been crucial in providing an invaluable resource in New Zealand with the patient samples. I've also had a newly developed association collaboration with Associate Professor Sherry Blinkeron, who's provided me with the breast cancer cells to do all of those experiments, and Dr. Kate Angel, who has helped me with a lot of the immunology background recently. But most importantly, one of the most key collaborations I've got is obviously with the Neurological Foundation, who granted me to be here, and the funders who actually make all the difference and allow this to happen in the first place. And what's really good is it's those two key collaborations I talked about before, the Neurological Foundation and their funders, that really help make us go that extra mile. And a good example of that is through some of the funding that is provided, I'm actually able to go to conferences internationally where I can communicate my research that I conducted here in New Zealand and show those pictures that I showed you today to clinicians and researchers from all around the world. Firstly, to teach them what we do here, but more importantly, to learn from what they do there, because often bigger countries have access to a lot more resources than us. And so the more we can learn from them and the more we can teach them, the better off we all are in developing therapies. And at that point, I'd like to thank you all for listening and for coming to the talk. Thanks also to our supervisors and advisors and all the lab group members that have helped me do this research. However, most importantly, I have to say thank you to all of the donors and their family whose, it's their donation that allows us to do the work that we do. And I hope that we can come up with therapies to help families in the future. And that's all from me. Thank you very much.
Well, thank you so much, Akshata. That was really informative and really interesting. And I really appreciate you taking the time to talk about the international effects as well, which is so interesting and so important to further that collaboration.
That's okay. Thank you very much. Thanks for having me.
So now we are on to the Q&A part of our session. So we've already had a couple of questions come through during your talk. So I'll just start firing these off if that's okay.
Sure, yeah, that's right.
So our first one, if you have cancer in another part of your body that migrates to the brain, is the brain cancer usually more serious than the other type? I guess I'm not sure if you can answer that.
Yeah, I can. Usually, yes. No, that's actually a really good question, and it's a really important one, because especially with something like melanoma, if you can imagine, your melanoma is on your skin, which you can see with your eyes, which means that a surgeon can get to it with their knives. So if you catch a melanoma early, where you see it in your skin, it's actually almost entirely curable. When it becomes really difficult is when it gets into the blood and then travels to different parts of the body. Now, if it goes to a more superficial part of your body, then again, maybe a surgeon will be able to resect it, remove it entirely, and then that means that you have a chance of that not spreading any further. So you can do that to any part of the body, but unfortunately, once the cancer gets inside the brain, there's only so many things you can do, because first of all, it's very hard to physically go into the brain to remove a chunk of it. Second of all, you can't just remove a chunk of the brain, because then you're not going to process. You need your brain for regular functions. So it is usually the brain metastasis, which is when the cancer actually goes inside the brain, that is the most deleterious time of the disease. And that's when you really have to, that's when the doctors get even more serious about these things. So the earlier you can catch it, the better. And with most cancers, that's why even with breast cancer and lung cancer, people try to prevent that from happening, which is why you have all these screening situations where you can do mole maps, where you do mammograms. All of those things are really, really important, because if you can catch it where it originates, great, we can get rid of it much, much, much easily. If it's gone to a different part of the body, we can try find it and get rid of it. Once it's gone to the brain, there's very little we can actually do to stop it from happening. Not to say there's nothing we can do, that's not true. There are therapies for brain cancer as well, but it's just a lot harder to fight, because the importance of the brain in just everyday function, you need the brain, right? So if there's something growing there, then it's stopping the brain from doing that. And then if you cut something out, that's also going to stop the brain from doing its regular. So yeah, that's absolutely correct.
Yeah, wow. Okay, all right, so why do these cancers target the brain when other organs might be easier for them to get into, or is that too simplistic?
Yeah, that's an interesting question, and there's lots of different theories. There's this one thing that we talk about a lot in metastasis, and people can look this up. It's called the seed and soil theory. It's sort of like a simplistic term given to it, and essentially it kind of talks about how certain cancers have a greater affinity to specifically want to go to the brain, and then once they're inside the brain, to specifically grow there. So while any kind of cancer can go inside the brain, we find that it's those three, the lung cancer, the breast cancer, and the melanoma, which specifically travel there. And it might, like, even though it should be difficult for them to go in there because of that blood-brain barrier, we find that they still manage to do it. And we're thinking there's a variety of reasons. Either it could be stuff that actually expressed by the cancer cells, which we're trying to figure out. But with melanoma specifically, one of the things that we've considered is, if you think about development of cells, and this might be a little bit complicated, but melanoma cells come from a lineage of cells that not melanoma cells, but melanoma is a cancer of what we call melanocyte, and a melanocyte is the pigment forming cells in your skin. So they're the things that make you tan when you go out in the sun. So they're there to protect you, but when they become radiated with UV, they become melanoma. And so those cells, when you're becoming from a fetus to an infant, those cells actually come from a lineage that brain cells come from as well. And what happens is that their father cell is the same, and then they develop and differentiate into brain cells, and then the melanoma cells develop and differentiate into melanocytes. And so we're thinking that it's because of that shared progenitor cell, that shared parent cell, that sort of allows the melanoma cell to really thrive when it gets to the brain, because it's got some kind of a parent environment that it's maybe used to. We're not sure if that's 100% true, and if that's the only reason, but we're wondering if it's because it's things like that that really cause some of these specific cancers to particularly go into the brain. And that's kind of the running theory at the moment. It's called seed and soil theory, if that's helpful.
That was fascinating. I had no idea about that. Thank you.
Yeah, sorry.
Okay, so I guess the next question is kind of similar vein. Why do some people's melanoma cells spread to the brain and others don't? Do you have any theories about that? Are there any theories out there?
Yeah, so it's actually really interesting because what we found more specifically with melanoma is that in some cases, there have been some sort of large-scale clinical studies done which show that you might not detect the melanoma in the brain once if a patient passes away because of the cancer. But usually when you get to, if you do like an autopsy, people have found that about 70 to 80% of patients who've had melanoma have had a metastasis in the brain that just wasn't diagnosed prior to them passing away. So with some of these cancers, I think the level of brain mets is actually underrepresented, if that makes sense, and that most of these cancers that preferentially metastasize to the brain end up in the brain eventually. With the question of why some might not, it might just be the stage of the cancer where it's at, in that for a cancer to get from where it is, which might be the skin or the breast or the lung, to the brain, it's actually a really inefficient process. There's everything in your body that's stopping that from happening, because it shouldn't. You've got your immune system and the blood vessel. You've just got the sheer blood flow. That's gonna, because it's so turbulent in your blood, it's gonna kill the cells as well. You've got the blood-brain barrier that's there to protect it. So there are a lot of steps that the cancer cell actually needs to overcome to get inside the brain. So it is likely that in some people that might just not happen. Maybe they've got a slightly less aggressive cancer, and that could be determined by the proteins it expresses and just how aggressive it is and how resistant it is to other drugs that it's being treated. Often it might be facilitated with other complications that the patient themselves have. If they can't tolerate drugs easily, then you can't give them stronger drugs, which means, you know, it's just small things like that. But at the end of the day, what we found is that cancers that like to go into the brain will end up in the brain. It's just a matter of when, unless of course they're treated with therapies, which is why we always say, if you're worried about something, get it checked. Absolutely get it checked, because it could be life-saving, absolutely life-saving.
Yes, definitely good advice. Take advantage of all of the screenings, those out there listening. So given that melanoma is so prevalent in New Zealand, which we do know, is our research respected worldwide as leaders here, or are there other countries in the world that are doing similar stuff?
Yeah, that's actually a really good question. Like yes and no, because we've got a very specific push. And when I say we, I don't only mean New Zealand, I actually also mean Australia in that, because New Zealand and Australia are both in sort of the same boat when it comes to the higher risk of melanoma. So we've got that specific push to really try and understand the biology of melanoma, but at the same time, I think we put more effort into melanoma prevention. So we've got a lot of effort that goes into the Sun Smart ads and things like that, which is even more important because we've got an environmental factor. So in that sense, we probably do a lot more in educating our population of what melanoma is, just in general. But in terms of the research that we're conducting here, we're pretty good. I think we're on par. We're not, like I said earlier, we don't necessarily have as many resources. So it's harder to, even just big machines, it's hard to bring it into New Zealand because we're so far away from people. So in terms of doing some of the larger scale, some of the more clinical work and some of the more animal model based work, those are probably more heavily done outside of New Zealand. Whereas at the CBR, we're probably one of the bigger groups here that are doing all the more specialized cell-based work. What's really important to note here is that we've actually got access to patient samples, which a lot of people might not do. So people might be doing really amazing research. Clinical work is totally different, but when you're working with cells or mice or something like that, they're working with a model that they've created to represent melanoma. Whereas we're working with cells that came from a patient who had melanoma, and that's from the Auckland Cancer Society. And if we're doing tissue work, we're actually working with tissues of cancers that came from a person through the Neurological Foundation Human Brain Bank. So that's a resource that we have here that nobody else in the world has. Well, I mean, they've got their own pools, but we've got a massive one over here, which is quite invaluable and very specific to the disease that we have in New Zealand, which is really, yeah,
absolutely. And as you say, an invaluable gift that those families and patients give research to be done.
But that's why the collaboration becomes really important, because if we're missing information from elsewhere, we really need to bridge the gap, so that you don't want to double up on the same thing. You want to help each other out as much as it helps.
Absolutely. Are there any things we can do to reduce the risk of the cancer cells getting into the brain? Is this something you've looked into?
Maybe it's the first thing is always prevention. So if you're looking at melanoma specifically, please don't go outside without sunscreen on, especially if this is not just for certain demographics. It's not just for Caucasian New Zealanders. It's not just for people who have a history of melanoma, although it's more serious in those groups. I have darker skin, but I would also always wear sunscreen before going out. And actually, we've noticed something that some of our skin of color populations actually end up with significantly worse melanoma, just because they don't think of it as seriously as people who, some of the ads might be tailored to are. So everyone should be taking care of things like that. With breast cancer, it's similar, doing regular mammograms. If you've got a history of it in your family, definitely keep an eye out for it. It is scary. It is scary to have to go and do that and then suddenly find out that you might have a tumor. But finding it early is the best thing you can do if it's there, and stopping it from happening is definitely the best thing you can do. With it getting into the brain, there isn't really anything specific that I can say you can go do this and this will stop your chances of it getting into the brain. But there have been some studies recently done, and this is very, very, very novel. So it's not, like a set, you have to do this, where people look at metabolic therapy. So essentially looking at things like your diet and having a healthier lifestyle, which sounds silly to say it that way, because cancer is cancer and it will happen if it's going to happen. But sometimes people say that doing these kinds of things like not overeating, trying to fast, trying to starve the cancer out essentially is a way to prevent these things. But they're very, very novel types of theory, and they're quite harsh. And usually you would never do that without getting recommendation from a doctor first. So I would say if you have a cancer and if you are worried, maybe go to your oncologist or your GP and ask them about whether something like metabolic therapy can help. So go like fasting and trying to, because the cancer cells need energy and glucose to replicate. So if you can do a kind of mediated fasting to prevent that energy from getting the cancer, will it help prevent its growth and migration? But that is something that your doctor would have to advise, because not everyone can do that. Fasting can also obviously hurt you as well. So you don't want to do that. So you would try and get advice from your doctors on that. But yeah, there's no obvious evidence of something that you can do that will stop this from happening. Unfortunately, not yet.
Yeah, absolutely. The key is the melanoma slip, slop, slap, and wrap, right?
Yeah, wear your hat, wear your sunnies. And after water, put it on again.
Yeah, that's right. Absolutely. Okay, I didn't. So here is our next question. So it starts off with thank you for the excellent talk. With your blood-brain barrier impedance model, is it possible to reverse the increased permeability by removing the melanoma cells? And then in parentheses, I realize this is not easy. Or is there irreparable holes made in the BBB by the melanoma cells?
Yeah, that's a really, really good question.
I have a feeling that came from a scientist.
Yeah, that's a fantastic question. It is. it would be very difficult to remove the melanoma cell from there because it's stuck. It's gone on. It would be very difficult to remove it while leaving the barrier intact in its place to then look at the certain areas of where the melanoma cell went in. However, I might be able to answer in a slightly different way, where we're looking at two things right now. One, currently the assay that I'm doing is on a static system. As you saw, there's a well, and I just put the two cells in. But that doesn't really replicate what's happening in your body, because in your body you've got blood flow. So the cell isn't in just one well, it's moving constantly. So what we're trying to do to kind of try answer that question is do the exact same experiment, but in a system that has blood flow, and not blood flow, but shear stress. So we, rather than having just a well where the both cells are, we'll grow the endothelial cells, and then we'll have the melanoma cells sort of circulate rather than put right on top. And I'm wondering, we haven't done that yet, but the idea is, just because building that system is a whole other thing. But what we're trying to see is if the melanoma cell still attaches and goes through. And then if it does, then maybe we can try and see if increasing or decreasing the flow reduces that. And secondly, we can try growing the brain endothelial cell in a 3D structure, which is what Sam, one of our students, was trying to do, to see if it then goes through and out. And if it's going down the other way, then it tells us two things. One, that it can go past and actually traverse, like in 3D, it can go through. Mm-hmm. And two, it'll help us to look at the melanoma cell after the cancer is already gone. Sorry, I said the wrong thing. It'll help us look at the blood vessel cells after the cancer is gone to see if the blood vessel seals up once the cancer has disappeared. So that's one way that we can check that sealing up. We have tried looking, we've put in other things other than cancer cell, like smaller cell types like T cells and immune cells that aren't so chunky and walking. When you add those in, what you see is a transient change in resistance where the blood vessels open up, the cells go in, and then the blood vessels close up again. So I think it is likely that those blood vessels are able to close back if they're not disrupted entirely, because we've seen some evidence of that with smaller cells, and so we kind of expect to see that with cancer cells if there was space for the cancer cell to go somewhere, which is why we're trying to build this 3D structure with blood flow and everything, which would also be better representative of your body as well. So I'm sorry that doesn't entirely answer the question, but if we could do it, if we could just pull out the melanoma cells, then we would, but just we don't have the system here to allow that. So we're trying to think of doing it in this other way where we can potentially look at it. Another thing to point that is we also think that the cancer cells target areas that are wounded in the blood vessel. So if you've got an area where you may be electrocuted a little bit, or if you try to I don't know, maybe it's just a weaker area in the body where maybe a one cancer cell already went through, it kind of looks like another cancer cell will use that same route. So we have some evidence to suggest that maybe they target weaker areas in the barrier. Yeah.
Okay, fascinating. Okay, so next question is, if the resection of the melanoma included lymph nodes, would this indicate a possibility that brain cancer might follow? It's a tricky one to...
So I'm assuming in this case, they're asking if there was a lymph node biopsy done with the surgery, I assume. If usually when people produce with aggressive cancers like melanoma, because we call melanoma malignant melanoma, and that's because it's known as an aggressive cancer. So your doctors will sort of preemptively try to do a lymph node biopsy to check if that melanoma has spread. So if you find evidence of melanoma cells in the lymph node, that suggests that it has potentially gone to other parts of the body outside of the skin. And at that stage, I think what the doctors will do is check the level of melanoma cells in the lymph nodes and how much of it is there, and then determine if there needs to be like a CT scan or something done to detect it elsewhere in the body. That doesn't mean that it's in the brain. There's not really any evidence for that at that stage, but it could be elsewhere in the body. And then it's up to the doctor at that point to do the right test to make sure that it's nowhere else. Obviously, if you wanted to check this in the brain, you would have to do an MRI, but it wouldn't, MRIs are quite expensive to do, and it would be hard to get on. We actually don't have that many MRIs in New Zealand, which is something that we're trying to change in the hospitals. But the idea is that you would do a CT scan, you would see if there's spread elsewhere in the body, and if that looks like there might be a lot of spread, then you would start progressing to look at other areas of the body as well. So you can always request your doctor, and often if the health system can't provide for it, I think you can ask to pay for it yourself to also get it done if you're really, really worried. But that's not always possible, of course.
Absolutely. Yeah. All right, thank you for that. We've got time for a couple more questions. So I think this one's quite interesting. For good cell wall health, to help prevent cancers, is it necessary to have a reasonable cholesterol levels? Is there
so cholesterol is, yeah, it's always going to be helpful to have better cholesterol health. Usually when we think about cholesterol, we think more about blood pressure. So we're thinking about blocking your blood vessels, which means that the blood flow through the blood vessel is interrupted or impeded or has added more pressure. And that's not a good thing, because that can cause, at the level of the heart, it's slightly different problems. But what you don't want is for smaller vessels to burst or because of that. I think that's unlikely to happen just because of cholesterol, and usually you get like aneurysms and things that happen in the brain anyway, just because of the high blood pressure that's in the brain, because it gets the first set, your brain gets the freshest blood essentially from the heart. It goes directly into the brain and then it goes everywhere else in the body. So in that sense, maybe that would be, because you don't want your blood vessel to rupture or be weak or have decreased blood flow to any part of the brain. That can cause issues just in general for your brain health. But if you did get a burst or something on top of that, then that wouldn't really be beneficial, because that just allows more things to get inside. Again, cholesterol wouldn't be the biggest impact of that, but it just helps to have better cholesterol just in general for better health.
Yes, absolutely.
Your heart.
Yeah, certainly. This one, I'm not familiar with this type of cell, so hopefully you are. I'm just wondering, does your research apply to other forms of cancer, such as Merkel cell? I'm sorry, I'm not sure about what Merkel cell is.
Yeah, so Merkel cell, I think, from my understanding, is an aggressive cancer, but it's quite rare in that I don't think we've got enough patient sample or patient data to say that those go inside the brain. So my research is quite specific to cancers that affect the blood-brain barrier and get inside the brain. And we don't really have a lot of evidence to say that it goes in. It's interesting that you asked it, because one of my collaborators, Associate Professor Sherry Blenkeron, is actually working with Merkel cells cancers as well. So I could ask her to send me some of her samples and just see if they have the same effect on the barrier and try it out. It's a good point, so I can test it out. If people are interested, it's an easy enough thing for me to do, as long as Sherry is happy to donate some of her cancer cells. But I haven't specifically looked at that at the level of the brain, and there isn't enough data, because it's not as common. There isn't enough data to say that it is something that travels, and then if it's not something that we know. I think it's more sort of primary, which is the issue with it, as opposed to secondary treatments.
Absolutely. Okay, we've just had this question come through. I think it's quite a nice one to end on. So it starts off with, thank you so much. Your enthusiasm is obvious, and I completely agree. What do you love most about your work?
Oh, that's a good question. I like doing the lab work, and the older I'm getting, I'm realizing is I need to have more students who I sort of interact with and then teach them about this as well. But I've said this before a lot in the neurological talks, and I'm going to say it again. But one of the most important things are these talks. Now, obviously today it's an online talk and I'm not actually being able to interact with anyone. But in the past, some of the ones that we've done for the public and also for academia, but academic people as well. But usually it's the ones with the public, often where there are patients in the environment as well. It's these talks that really help ground us about what we're doing, because you kind of forget sometimes. You forget the importance of what you're doing in the lab, because it becomes, you're looking at plasticware and cells, and you can't really easily relate that to, what the disease is currently. So because we don't have that clinical input, it's actually these conversations where we're sitting here talking to people, seeing their reaction and seeing the difference that it makes, that is really helpful for me. So I really enjoy that, and I wish that we could have done this in a proper live environment where we could actually engage with each other and have conversations. But it's one of the key things, and I've been able to do this a lot through you guys, through the Neurological Foundation, because you have so many of these talks. But I enjoy it the most, and it's sort of one of these things that allows me to then go back into the research and say, that's right, I have to do this. And I've gotten like 2 extra ideas from this talk. Like the people from the general public have given me 2 extra ideas that I can go and test in the lab now, which is great. So this is probably my favorite part, to be honest.
Yeah, that's amazing. And again, with the collaboration and connection, it's so important. Well, and on that note, Akshata, we're going to let you get back to your very important work, and I'm going to hold you to wanting to do a live event. So everyone who's watching, keep an eye out for when we do book Dr. Anchan for an in-person event sometime soon. So thank you once again for all that you're doing and for sharing your work with us. And everyone at home, thanks for joining us. This recording will be available end of the week or early next week. And we look forward to seeing you at one of our events again soon. Thanks very much.
Thank you. Bye-bye.
Bye.